Okay, to start my class. So today, first thing which we have to do in this practical is to retrieve DNA sequence from a database as per your syllabus. So what you are going to do in this aspect is first check your WhatsApp today. See that I have sent you some practical files, some sequences, huh? And there you will find lots of uh, interesting things which you can use later in life. And for example, you have to do your practical copies also. So that is already given. How you will do it, I will explain as we go throughout in this practical. I'm sharing my screen now. Okay. How to first access a DNA sequence? Say you are gone for some research work. Your supervisor says, okay, please bring out the sequence of E. coli 5S ribosomal DNA sequence. We need that sequence for your study. How will you do? Where will you search? You must be knowing all these things. So how do you do it? You do it like this. You go to Google or Bing or whatever, go to the NCBI site. And the web address is ncbi.nlm.nih.gov. It's there in the practical notebook. Only what we will do today, I will take screenshots and this I'll send you for your practical book. You did not follow your seniors' copies. So now I have opened this site, ncbi.nlm.nih.gov. So in this particular website, you will see that this search box is absolutely on the top. And on the left side, you'll find all databases. So what is DNA? So there are lots of things: bio collections, bio projects, bio sample, books, cleanware, conserved domains, lot of things. But what actually your boss has told you to do is your boss needs the DNA sequence of 5S ribosomal DNA. It will eventually get transcribed to form 5S ribosomal RNA. So what you do is you have to find out where will you search. So what a DNA sequence or RNA sequence, it is a nucleotide. So you go to this nucleotide over here. It's absolutely free of cost. You don't need any money. This is available on the public domain. Even when you are doing research, you get new sequences. It is advised that you go to PubMed and submit your sequence. So you go to this nucleotide and you can submit your sequence. But let us talk about retrieving it. Somebody has done the sequencing and now you have to retrieve it. You have to retrieve it back. Sounds tough, I guess, but it is not at all tough. All you need to do is go to this particular website. On the top, your supervisor has told you, retrieve me 5S E. coli ribosomal DNA sequence. For example, it can tell you even to retrieve histone sequence, heat stress transcription factor sequence. All the process is same. You go by the same manner, and again, gene after gene, you can retrieve from here. So you go to nucleotide, keep it. So what he has told about 5S ribosomal DNA sequence of what? E. coli. You just simply type that and you search on NCBI. And now what do you see? You see a lot of things coming. You find lots of matches, lots of things coming over here. But the most relevant match is, can you see 5s ribosomal DNA? Clear? Doesn't matter. Ribosomal RNA, but what you get is the DNA sequence. Normally, you call it as RNA. And then you search. And this opens up. This particular website opens. And in this particular website, you will get all details about this particular DNA sequence, which you normally call it 5s ribosomal RNA. I need the DNA sequence of 5s ribosomal RNA. That is what I need. Okay. And you can see all the details about it. What you have is where locus number. You see this number, this is very important. You always copy this number. For example, I have already done this work. I had copied it over here. It was another from another accession number, but it has been copied over here. You can use whichever, you can use what I've given you over here. You can use this, or you can do it on your own, and you can do this search on your own. I would request you people to do this search on your own instead of copying and pasting everything. It is very easy. So take a printout of this page. Okay. Now I'm going to the previous page. Take a printout of this page. Especially, rather more than the printout, take a screenshot, I would say. A screenshot about this region. What you are seeing on the screen, take a screenshot. I've taken a screenshot for you. I send this to you. Okay. So I'm again taking a screenshot from this. So I'll send these screenshots to you. Paste this on your lab notebook. So we have come, and here you don't need any screenshot. So you take a direct printout. Below, all details, which publication, origin, everything, whatever paraphernalia you need about whatever you want to know about this gene. Where can you get details? For example, this accession number is very important. You know that this was the Sakai strain. Sakai is a particular E. coli strain. They took the sequence over here. You will find lots of other strain E. coli also. They will have different accession numbers. But whatever DNA sequence you are giving, please make sure that you give this data to your boss also. That at least this NR075284. This is the locus, this accession number, reference sequence. You always give this sequence to your boss or anywhere in your or in any publication. Whatever you are doing, you need to give this sequence. So this is the most important thing. Keep it in your mind that this sequence number always has to be there. It is something like I always used to say, whenever you're writing scientific name, you have to underline it. The genus name starts with a capital letter. species name with a small letter. It has to be underlined. You have to write all these things. So you have to be absolutely certain what you want to do. Then you have authors who found out this thing and the title of the paper: complete genome sequences of hemorrhagic E. coli. So this is a very pathogenic strain. It affects your stomach. So in your stomach, it causes ulceration. This particular E. coli strain, because entero means inside your gut. Hemorrho means there's a hemorrhagic strain of E. coli. So this causes bleeding in your gut. So this is not a good strain to have in your E. coli. There are harmless strains. So this is a very pathogenic strain. Clinically, it's a very significant strain. Therefore, this came out first. So they have done it, strain K12. Which journal? You can find this original paper if you search. Authors are there. And there are many other details about authors where you can find everything. But at the bottom end, you find this DNA sequence. Each of these 10, this is actually 10, each block of 10 nucleotides. TGCC, TGG, CGG, you see all these things. This is the DNA sequence. Now you need these DNA sequences. It's absolutely essential that your boss has told you, otherwise he will kick you out from his lab. So you have to do this. So how do you give your boss? You take a printout and give it to your boss. Your boss will say, what will I do with so much? I need just the sequence. Why are you giving me all this additional junk? So what you need to do is you need to open a notepad. This is my favorite thing. So here I save this notepad as E. coli ribosomal RNA DNA sequence. I just saved this notepad. You can use Word, you can use anything, whatever you want. You do it slowly, watch this video, do it step by step. And it is not going to be very difficult. So on the top left side, I'm taking a screenshot. I'll give it to you later. On the top left, in this region, there is a word called FASTA. The left side where you can see here, it's called FASTA. So what is this FASTA? FASTA is a special type of sequence. The sequence which you will give your boss, the sequence which you will save in your notepad, the sequence, the format in which you will save everywhere. This is the format you will use in your bioinformatic research. This is the format which is generally used. Even if you are giving it for sequencing, you have a FASTA instead of FASTA, you have FASTQ. This is a DNA sequence. But FASTA, everything is in FASTA. So you click FASTA. Now, what is this FASTA format? So you can see this is not the part of the FASTA sequence. This is not FASTA sequence. The one which I've highlighted in blue is not FASTA sequence. The FASTA sequence consists of this. Most important is you have the greater than sign over here. After that, you write about the ribosomal RNA strip. Okay. What it is. You don't need to write so much. Just enough information that you can identify which sequence you have found out. So you have this, you can give this, you can take this E. coli. I can take the whole thing also, it's no problem. But remember, equal to a greater than sign, some details of the sequence, it can be small, it can be large, and all the sequence written one after the other. So I copy this and I paste it. This is a faster format. Okay, this is how you retrieve a E. coli DNA sequence. So let us keep this saved. So this is the first experiment, first thing which you have to write in your lab notes. Retrieving a DNA sequence from the NCBI database as it's given. This is the first experiment. And all those screenshots which I'll give, and this one, I'll take a screenshot, I'll enlarge it. You don't need to take individual, you put them in one big file, all of these little screenshots, take one printout. Otherwise, it becomes very expensive. So this is it would have been better if I gave it in the other way around. But anyway, you can paste it yourself also. Whatever, you take this little bit region. I will send this sequence also. So I'm keeping this now. This is saved, has to be saved. Next thing is we will see what is blast. Inside this Google search, the same Google search which we did, you will find another thing known as blast. It means basic local alignment search tool. Now, what this blast will do, it is going to find out sequences in the database which have got similar sequences. So you have an unknown DNA sequence. In this case, we have a known DNA sequence, but you have an unknown DNA sequence. So what you are going to do with this unknown DNA sequence? So you don't know, say for example, here you don't know what this particular sequence is. Okay, you have this accession number. You need to do blast. So you go to this blast. So this is the site for blast. I'll take a screenshot of blast. So BLAST means basic local alignment search tool. So what it does, BLAST finds regions of similarity between biological sequences. The program compares nucleotide or protein sequence to sequence database as calculates the statistical significance. So essentially, you bombard a DNA sequence, unknown DNA sequence, you have no idea about it. You just bombard it into the database, just paste and blast. Give it a blast. So it's going to search for local alignment, similar DNA sequences, which have got lots of small similarities, little domains which have got similarities present. This is pretty exciting in the sense, and this is a very useful tool. Anywhere you do research, the first thing you need to learn is BLAST. Bioinformatics thrives on BLAST. People are putting all the sequences in the database, and now you have an unknown sequence which has come to you. You have sequenced a new organism. Suddenly you are walking on the road eating your egg roll or mutton roll, and then suddenly you see earthworm or some new species of worm walking on the road. You pick it up, take out its DNA sequence, and now what is the species? Is it known or unknown? You don't know. For you it is new. Now you've sequenced that DNA, and now the DNA sequence has come, and now you want to know what is this. So how do you know? You'll blast. Even if you have found out a new novel organism, you're going to get you have a very bright scientific career, definitely. But what you will also get to know is what if it's a new organism, that's fantastic. But what genes, what are the different genes, what is the type, what are the molecular details, what are the different types of genes present inside it? Is it does it belong to the phylum Arthropoda? Does it belong to the phylum Echinodermata, mollusca, whatever? There are all worms present everywhere. So what is it? Or is it a brain worm? We don't know. So whatever it be, this is very important that you learn it. So now we are going to do a blast. So since you have got a nucleotide sequence, you will do a nucleotide blast. But you have a protein sequence, you do a protein blast. You have a nucleotide sequence, you do a nucleotide blast. Now I don't know. This is nucleotide sequence. I go to nucleotide blast. If you people have questions, please do interrupt me. And now you can see enter query sequence in FASTA format, or you can give an accession number. What is the accession number? I've shown you. This is the accession number, the 075. You can give this number because it's already in the genome, but you don't have any accession number. Say, for example, this is an unknown DNA. You have no accession number. You simply copy. You copy the sequence and you paste the sequence. But you have to give it in a FASTA format. Be careful. FASTA format will have a greater than sign. Let us say this is unknown DNA. We don't know anything about this DNA. Although we know it, but still, imagine हम लोग के पास कोई information नहीं है इसके बारे में. Now you need to know which is the DNA. This is a detective work which you are going to do. You are going to find out what this damn sequence is. Let's find out. So you can upload a file also. You can upload the text file which you have. You can upload it from here, but it doesn't matter. This query range, maybe someday later I will explain, but for your exams and purposes, let's not think about it. It's nothing. If you count all these DNA sequences, you only want to search from, say, after 10 nucleotides, from nucleotide 10 to nucleotide 20. You just write 10 and 20. That's going to, so only search, only that will be part. The other part is not going to be searched, right? Now, you go for the standard database. You can check for different databases also, but you do it for the standard database. And let me warn you, right now it's 3:32 and America is awake now. Once America is awake, this is very slow. This site is very slow. So if you are a PhD student, I would suggest that do all these things in the morning when America is sleeping. But because once America, to a certain extent, Europe, especially in Switzerland, once America is awake, East Coast, West Coast, all Central region, that's it. Once it's awake, it's very slow. So right now we can expect the time right now. It's 15:30. We are still in during the daytime. So in America, it's early morning right now. So America is sleeping. Maybe we can find it to be faster. Let us see. So we have checked out. It's a nucleotide collection. Okay. So I've taken a screenshot, right? So now I've pasted it. Now without this. This is the general blast which I'm showing. Yeah. I would prefer to show results in a new window because you want to see what you have done over here sometimes. This is how I actually do it myself. Now you can select, you can optimize for highly similar sequence. That is most similar sequence. It's perhaps the same species also. You can do for more dissimilar sequences. Some sequences, the regions in the sequence may not be good, may not be similar. And you have somewhat similar sequences, last 10. So this particular, I'm going for highly similar sequence. मुझे तो निकालना है ये क्या है DNA first of all. We don't know anything about it. And you do a blast. It's opening in a new window. It's still, the job title is going on. It's searching, and here you have come, the result has come up. Okay. Now, you don't know unknown DNA sequence. Okay? This is what you're searching. Everything, you don't know anything about it. So let's see what it can be. Dekho, what comes out actually. First, let me take a screenshot before I lose it. Now, what comes out actually, मैं इसको बड़ा कर रहा हूं. Sequences producing significant alignments. ठीक है? Sequences producing significant, मतलब alignment मतलब x के साथ x मिल गया, y के साथ y मिल गया, a के साथ a मिल गया, g के साथ g, जिसे बंगाली में कहते हैं खापे खाप पंचुर बाप, right? Everything should be same. So the first hit, 100%. There was some, and here you have the E value. This is the statistical score. This is 1 to the power minus 53. There is the chance that there is an error. Almost negligible. 1 to the power minus 0.00053 times जाके तब maybe one mistake हो सकता है. It's 100%. 100% एग्जैक्टली बोल नहीं पा रहा, थोड़ा सा कम है, बट 100 ही है. So you get the complete DNA sequence. मतलब डीएनए में कि सब इतना सब 100% कहाँ से आ रहे हैं? सो मेनी 100%, so many 100%. मतलब the same strain, different people, maybe from the same lab, एक ही चीज बार बार वो लोग ने सबमिट किया है, ये नाम देके. So they have taken the same thing, the same sequence, maybe the same organism. E. coli, मेरे पेट में जो E. coli है and this इन लोग के पेट में जो E. coli, more or less they are same. This genetic sequence, few changes there may be, few changes, but 100% hit means it is supposed to be E. coli. Now you understand. Oh भाई ये तो E. coli का DNA है रे. You can even click on this place. If you click, query sequence, that is your unknown sequence. ठीक है, दिस इज योर क्वेरी. एंड सब्जेक्ट मतलब जो निकला था ये इकोलाई वाला भाई. यू गेट, यू सी, पूरा सीक्वेंस अलाइन करके 100% मैच आ गया. जहां भी जिस तरह से भी तुम करो पूरा अलाइनमेंट 100 आउट ऑफ 100 मैच आ गया. एंड यू वांट टू नो द डीएनए सीक्वेंस आईडी, यहाँ पे दे दिया. ओपन दिस. अच्छा, यहाँ पे भी जीन बैंक, यू हैव गॉट जीन बैंक. You can have all this is DNA, whatever it is, and this type of alignment, it is एक के साथ एक का जो अलाइनमेंट होता है, दिस इज नोन एज पेयर वाइज अलाइनमेंट. दो सीक्वेंस, one is your query sequence or a one you have selected. Maybe you have got two DNA sequence, one known है एक unknown है. You are thinking the same, you can align them too. Align करेगा, it will be same. टी टी जी जी, अगर इसमें एक न्यूक्लियोटाइड डिफरेंस है तो यहाँ पे एक ब्लैंक आ जाएगा. तुरंत यू विल गेट टू नो दैट देयर इज एन एरर ओवर हियर. एरर नहीं, देयर इज अ गैप ओवर हियर, देयर इज अ मिसमैच. द वर्ड इज मिसमैच. एंड योर स्टैटिस्टिकल स्कोर थोड़ा सा कम हो जाएगा, इट विल बिकम लेस, अ बिट लेस. That is the beauty of this system. Let's get back to the graphic summary. जितना भी सीक्वेंस है कंप्लीट रेड, मतलब सब सबका सब लग गया. This is what you got. And next you have got alignments. This is pairwise alignments you have seen. You can go to taxonomy. You can, यार ये तो इकोलाई है, एंटेरोबैक्टीरिया है, पेट के अंदर वाला बैक्टीरिया है. हाँ, that is pretty good. You get all the details over here. And this 100, you can even set, I want 100, I want 10. Huh? Show 100, show 50, show 10. 10 करके I'll take. 10 करके ही लो, 10 करके I'll take the print out. First this part. Next नीचे वाला part. So you get the whole overview over here. This is absolutely very interesting. Currently, my focus in my personal research, I do a lot of bioinformatics. Of course, I need to do these blasts, even though it may seem very basic, but still you need to do these things. Next, in the next class, which I will be talking about how you make phylogenetic trees, how you use cluster, and how you make phylogenetic trees. So in this manner, what I've already done, in the before the next day is I have E. coli 5S RNA DNA from S aureus also I have taken from the database. Similarly, I have taken out the nucleotide sequences. Then from K pneumonia, I have also taken the 5S ribosomal RNA sequence. From Mycobacterium bovis also I have taken the sequence. From Salmonella enterica, so I have used it. I've already taken, right, like I have taken the sequence over here. Instead of E. coli, I typed S aureus 5S ribosomal RNA DNA sequence. आ गया. Accession number, I have done blast and all everything like this. मतलब blast करने का जरूरत नहीं है. But I've taken the DNA and सबको यहाँ पे बिठा दिया. ठीक है. Next day, what we will be doing is we will go to this website called Clustral. And ये सब सीक्वेंस को मैं कॉपी करके बिठा दूंगा. In this Clustral W. This will align your sequence, multiple alignment. That means I have 5 sequences of ribosomal RNA. One after the other, they will be stacked and aligned. You will see which nucleotides are similar, which are not similar, which are missing. Maybe one ribosomal RNA sequence of E. coli is larger than that of S. aureus. एक का है 100, एक का है 80, 20. You will see it will be there will be a gap. You will see what the similarities are in all of them. And you place them the FASTA format as they say. एक के बाद एक FASTA format बिठा दिया. Greater than sign something written नीचे sequence. फिर उसके नीचे greater than sign something written sequence करके 5 बिठा दिया. And you will execute multiple alignment and the sequences will align. A particular type of file called Philip file, PHP file will come. That particular file, you are going to use that particular file and you use it in a software known as Philip. You can do it also in Python programming, which I normally prefer Python programming to do this because if you do it in a particular Python or any R or any sort of computer programming language, here, for example, today I have got says, for example, 100 people have submitted a sequence here. So today I have done this experiment. This is an experiment with a retriever. I have done a bioinformatic experiment. Even if it's a small experiment, it's a still significant experiment. आज ये experiment किया. This result which I have, I need to reproduce it 3 times, at least, if I want to publish something. But so today you have done something with 100. Maybe 1 month later, तुम्हारा बॉस बोला कि और एक बार कर लो तो भाई ये यही तो आ रहा है. छापने के पहले, गलत छापने के पहले थोड़ा सा वेरीफाई कर लेते हैं. सो व्हाट टू डू नाउ? आई गो इसके बीच में विद इन दिस 30 days और भी 150 का सीक्वेंस कोई सबमिट कर दिया. नाउ इफ यू डू दिस, it's going to compare with the new 150 sequences. Your values which you have got. So suppose this E value क्या दिया? 10 to the power minus 53. उस समय और भी क्लोज सीक्वेंस आ दिया, 10 to the power minus 58 चला गया. What will you do? So you cannot reproduce it exactly. Maybe you will get the right. तुमको देख के हाँ सही है, but you cannot print it, publish it. So if you do it in this blast sequence, in this Python programming, in your particular code which you will design, you have already put in the parameters. All you have to do is to run that code. You get the same reproducible data again and again and again. So therefore, nowadays I don't prefer doing all these things basically online because it can change. But to show or to get an instant result, something like this, it is very difficult. So already you have taken a database, you have already made a code, you have retrieved all the sequences. तुम्हारे अपने पास retrieve है. It's attached to the code. The code is displayed in front of you. Nobody can challenge you that is wrong. तुम लिए हो the code, you cannot change those things. कोड के अंदर लॉक इट, दैट इज देयर, सेफ हो गया. अब तुम अगले बार तुम करोगे उस कोड को ही रन करोगे. बॉस बोला थोड़ा सा पैरामीटर चेंज कर दो, जस्ट मेक इट लेस स्ट्रिंजेंट, ताकि we can get more similarities or more make it more stringent, that we can get less बहुत ज्यादा similarities का, जितना कम similarity उतना अच्छा है. Want to see the difference, not the similarity. Depending upon what your research idea is, what you're thinking, you can actually go ahead. Okay, so I will stop my class here. So next Wednesday इसी टाइम में यहीं पे सेम प्लेस सेम लोकेशन. ठीक है, तुम लोग का भी एग्जाम आ रहा है, तो आई एम नॉट कॉलिंग यू टू द कॉलेज. नेक्स्ट वेडनेसडे घर में बैठ के ही ऑनलाइन में ये कर लेना. ठीक. So any questions?